So good morning, good afternoon, everyone. I'm Paul Danis with the consortium, and we're happy to have you here today on the March edition of Proteoform Thursday. Today we're lucky to have Jake Melby with us from AstraZeneca, and he'll be giving us a great talk today all about some of the challenges. Real sensitivity is a big issue in top-down. Everyone wrestles with that, so it'd be great to hear some of the insights Jake's going to have about this. Just a quick little intro. We definitely want to thank our sponsors who make these kind of events possible. We're happy to have Sear, as well as Thermo Fisher, Brooker, Cyax, Lilly, Agilent, and Pfizer supporting our activities. So if you see those folks or if you're part of those organizations, we thank you and appreciate your support going forward. Just one thing I want to give is that some of you may know we have a native top-down initiative we started a couple of years ago. Frederick Lermite from TU Darmstadt's been leading this. And the whole idea is to try to overcome some of the challenges of top-down methodology using native. We know it's challenging to start this kind of technology up. It's tough to compare across laboratories. So Frederick ran this study where the idea is to develop and test some protocols across numerous laboratories for both protein complexes as well as native proteins. And then come up with some general guidance that we can have and provide to the community. And this has been submitted to Nature Methods, been reviewed, updated, and the final submission's been put in for the title is Top-Down Mass Spectrometry of Native Proteoforms and Their Complexes, a Community Study. So hopefully that's out shortly in Nature Methods. And in the meantime, there is a preprint on Research Square if you want to have a sneak preview of what's coming there. So we hope to have that out to you. We'll let you know when that's available, because that should be really interesting to see for a lot of groups. The other thing is we're still open to speaker suggestions for Proteoform Thursday, and these can be, early career people, or they could be mid-career people. We're really open to a lot of different types of things. So if you have someone you'd like to hear about, have them tell their story, or if you'd like to tell your own story yourself, you can either put a comment in the chat to me, or you can send an email to the office at tdprot.org, and we'll be happy to get you on the docket here and have you present your story. So thanks again to all these great speakers, and we're going to add one more to that list today. So let me hand it over to Jake Melby, the Analytical Sciences Group of AstraZeneca in Maryland, to talk about novel strategies to address challenges of sensitivity in proteoform, top-down proteomics. Go ahead, Jake, it's all yours.
Thank you, Paul. Thank you, everyone, for coming. So as Paul said, the title of my talk is Novel Strategies to Address the Challenge of Sensitivity in Top-Down Proteomics. I included this I'm in Gaithersburg, Maryland, which is just outside of DC. So I included this nice picture of the cherry blossoms down by the National Mall. I didn't take this, but they are in peak bloom right now. So lovely place to be. So just a little bit about my background before I jump into the actual research talk. I went to the University of Pittsburgh. I got a bachelor's in science of chemistry, and then I worked for a company that made quadrupole mass specs for about 2 years as a service engineer. And after that got a little tiring, I moved over to the University of Wisconsin in the analytical chemistry PhD path, where I joined Professor Inga's lab, hence the reason why I'm talking to everyone here in a top-down session. I also had a chance to do an internship out in Merck in South San Francisco, where I was doing some middle down of antibody drug conjugates. And since about this time last year, I've been over here at AstraZeneca in Gaithersburg, Maryland, within the Analytical Sciences Group, which is a late-stage characterization group. So I wanted to I know this is a top-down section, but I kind of wanted to get into some of the real history and some of the beginnings of the top-down field. And kind of the start of all of this and start of much of bioanalytical mass spec was the invention of electrospray by John Fenn, published in 1989 for large molecules. The paper from Joe Liu and others at PNAS kind of demonstrating top-down of proteins within the gas phase using ESI. The McLafferty group and Professor Kelleher, this paper in JAX in 1999, kind of defining top-down proteomics versus bottom-up proteomics. And then if you jump ahead several years, the consortium was started in 2012, and the proteoform paper that has been cited, countless times was published here in 2013. So all of that brief history of just the beginnings of the field, for me to kind of get more to the point of, what is a proteoform and why do we have a whole consortium kind of dedicated to characterizing these things. So a proteoform is going to be defined as all the different molecular forms that can come from a single gene as a result of things such as alternative splicing, post-translational modifications, and mutations. And the reason why this is so important is because of the fact that it's really at the proteoform level that things are implicated in a disease setting, and very much in the industry that I'm in. It's really proteoforms that are kind of doing all the action. Excuse me. So to put this in like very kind of layman's terms, at the very beginning, when the field was kind of starting out, you saw a lot of top-down of single proteins, simple mixtures, and then we moved on to doing top-down proteomics of complex mixtures. And now we're to the point where top-down is being used to study proteoforms in a disease setting. But come on, slides. There's still a lot of challenges that remain in top-down in order to kind of get it to a state where it's being used in all settings. So back in pandemic times, I was thinking about this, making these slides. Me and David Roberts and all the other co-authors on this paper had a chance to write a review on the challenges of top-down proteomics to kind of celebrate Ng's Beeman Medal in 2020, which was an honor to write and a lot of fun. But things that still kind of hamper the field a little bit, and things that Ng's lab has worked on include things such as protein solubility, the dynamic range of the proteome, connecting proteoforms with function, finding better data analysis strategies, addressing the challenge of throughput, as well as the proteome complexity. But kind of where I jumped in 2018 and up until last year, I was working on the challenge of sensitivity, which kind of stems from, the fact that as proteins get bigger, the signal to noise is going to exponentially decay. And this is a multifaceted problem in terms of sensitivity, because things that are smaller and higher abundance are going to be easier to detect. And also the fact that, as proteins get larger, you get more isotopes, and kind of all of top-down is fundamentally hampered a little bit by sensitivity compared to the bottom-up field, where peptides are kind of at a perfectly ionizable state. So there's still a lot of challenges that remain. And all of these challenges that I already had mentioned are compounded even further when you try and do a high sensitivity experiment by top down. So because of all these challenges listed, most top down studies that where you're trying to do deep proteoform characterization use quite a bit of starting material, specifically if you're doing multidimensional chromatography. So my talk is going to kind of focus on two cases where we came up with a strategy to address the sensitivity challenge. The first of which is a paper that we published in PNAS around this time last year, where we did top-down proteomics of single muscle cells. So before I jump into that, I want to give a little bit of background on skeletal muscle itself. So it's a highly abundant tissue in the body. I think it's the most common, actually, and it enables a wide range of movement, posture, and balance. It's really heterogeneous, as you can see from this drawing here. It's made up of tendons, nerves, blood vessels, and then at the very crux of it are these things called muscle fibers, where they're more or less bundles of the sarcomere, which is the contractile apparatus of muscle. And several papers, many of which came from Ng's lab, show that post-translational modifications and isoforms of these proteins have profound effects on the muscle functional properties. So it's really again, to emphasize the proteoform that is, controlling this muscle movement instead of just, protein abundance. So during my first year, actually, I had a chance to kind of complete a project with a former student, Yutong Jin, where we used top-down proteomics to study 7 different rat skeletal muscle tissues. And we were lucky enough to have this as a journal cover in JPR. But basically, the idea here was we took these 7 different tissues, performed a 2-step extraction to get the contractile proteins, and then performed online LC-MS/MS to characterize these different muscle proteins. And as you can see from these different chromatographic traces, all of the muscles have some unique traits to them. And when you actually perform the online MS/MS, you're able to unambiguously characterize these protein isoforms that have high sequence homology. But when you're doing this at the intact protein level, you show that you're unambiguously characterizing these different isoforms. So that was all well and good, but kind of from the background that I just described is that skeletal muscle is a really heterogeneous mixture of things, many of which you'll find different fast twitch and slow twitch muscle fibers within the same muscle. You can see from this SDS page gel that there's a lot of different proteins that are found within muscle. And we were kind of thinking, this study was using 5 to 10 milligrams of tissue, which is, not a ton, but it's also quite a lot. So we were wondering if it was possible to do this at the single muscle fiber level. So just to give more context as to like why this is important outside of just, a sensitivity experiment, single muscle fibers are multinucleated single cells. So there's fusion of multiple precursor cells that produce, relatively large cells when you're comparing it to something like a HeLa cell. And I keep saying fast versus slow twitch. That's largely how the muscle fiber itself is classified, and it has to do with the isoform of myosin heavy chain. So fast twitch muscle fibers are used more for anaerobic metabolism, kind of, liken it to a sprinter, whereas slow twitch muscle is used more for oxygen, uses oxygen for fuel and is more for longer duration type exercise. So most muscles are going to contain a mixture of these muscle fibers. So if you're doing it at a tissue level, you're getting an ensemble measurement of all of the different muscle fibers in that piece of tissue. So because it's a relatively large cell, because it's heterogeneous in nature, we kind of hypothesized this could provide a proteoform rich system to study by top-down proteomics. And this is also to give context how small these things are. There's microscopic images that kind of show that these are usually about 1 to 2 millimeters in length and then maybe 100 micron in diameter. So, I mean, I'll get into it more, but they're really small. Just to kind of hammer home this point of why single cell omics has become so popular and what the motivation is behind it, I always appreciated this paper in Cell that kind of demonstrates if the dotted line is the measurement and these are populations, it can show you that an ensemble measurement at times can do a pretty poor job of trying to capture a representative view of what's going on in that system. So the same idea works here for single cell proteomics. So it's really important in heterogeneous cell populations, such as a tumor microenvironment, tissues, or even cell culture has been shown to have cell to cell variation. So the way I've always thought about this is the difference between drinking a fruit smoothie and eating a fruit salad. So when you're having a fruit smoothie, which would represent, bulk omics, bulk proteomics, it would you would kind of be tasting all of the fruits that are within it at the same time. Whereas if you're having a fruit salad, assuming you're eating it piece by piece, you can kind of um, taste each of the fruits, which is more likened to single cell omics. But while the single cell genomics and transcriptomics field has kind of progressed pretty far, this is largely due to the fact that they're able to do PCR. And within a single cell, the protein content of a cell such as HeLa is around 250 picograms, and we have no way of amplifying that material. The proteome is also far more complex in terms of a measurement. There's abundance differences, post-translational modifications, and then even the fact that measuring a nucleic acid versus an amino acid, there's fundamental differences in how that is done with current technology, where for genomics and transcriptomics, you're using a fluorescence detector, which is highly sensitive, and you only have 4 bases to measure. When we're trying to measure an amino acid, you're now dealing with up to 20 bases, and the detection method is using a mass spec, which is not going to be as sensitive than fluorescence. So that kind of gives some context as to why, the single cell proteomics field is lagged in terms of development time compared to the other fields. So I was a first year grad student, I think, first or second, when I started to work on this project, and I kind of went back to the drawing board and with the fundamental goal of how do I get this single muscle fiber, get the protein out of it, and get it into the mass spectrometer. So kind of leaning on some of the other literature that was in the field, such as the group in PNNL, where they're using the nanopots to kind of minimize sample losses by doing the whole preparation in a droplet. I, kind of took from that sample preparation is really important, and minimizing the number of steps and handling of your sample was going to be very important. So we basically looked at every step in this workflow, from sample preparation to cleanup for mass spec, to the actual liquid separation mass spec method, and then also tandem mass spec to try and maximize the amount of protein ions that we were getting from our sample itself and into the mass spec. So something that was pretty important through this development was a collaboration with the team at Nuomics. And I'm just showing some of the biggest things that influenced this study. There's plenty of other sample prep type stuff that's more in the SI of the paper. But this was really important in terms of like really big differences. So when you're looking over here to the right, this was just using a conventional ESI sprayer, whereas this was using the Nuomics source. So just to give more background on what the Nuomics source actually is. So it takes microflow flow rates and splits them into 8 nanoelectrospray emitters. So you get the benefit of microflow separation, which is really nice for intact protein separations, with the sensitivity of nanoelectrospray. And using this source and using a standard protein of carbonic anhydrase, we were able to get down to a 500 picogram detection limit of carbonic anhydrase. Sorry, I had a tickle. So this is kind of the first figure or main figure in the paper where it goes through the entire workflow that we developed. So we're starting with a single muscle fiber isolation, which it has to be done under a microscope due to how small these things are. We first, with a collaborator, measured some functional measurements such as twitch force to kind of see how the muscle actually contracts, which is also a functional property of the single fiber. We were performing a lysis with HFIP by freeze thaw, something that we also had seen in the literature from the Slavov group. To basically, and HFIP is a mass spec friendly solvent. It has extensive hydrogen bonding. So essentially, when we were able to get the fiber into the HFIP and perform freeze thaw, it lyses the whole single fiber. Once we had that sample, we loaded the entire thing onto our LCMS system. We're using a thermo map pack column with 150 micron ID column, so the flow rate was very low at 2 microliters per minute. And this was coupled to our pneumomic source, sprayed into a Bruker Maxis QTOF, and then all of the data analysis was performed from there. So we wanted to pick single fibers from 3 functionally different muscles. So the 3 that we picked was vastus laterales. I'm just going to say VL from now. This is mainly composed of fast twitch fibers. Plantaris, or PLN, not phospholamine, sorry, Holden. Soleus, which is SOL. So the soleus mainly is composed of slow twitch fibers, and the plantaris is a mixture of both. So this is to go over the functional data itself. As I already kind of mentioned, we had these 3 different, or fibers from 3 different muscles, 10 of them for this measurement. And then they were dissected and more or less had their functional properties measured by our collaborators lab in the form of a maximum shortening velocity. So essentially what you see here is that at a functional level, so just measuring the speed at which the muscle changes during a contraction, you can see that these single fibers all had different properties, with the soleus with mainly slow twitch fibers having the lowest velocity, and the VL with mainly fast twitch fibers having the largest maximum shortening velocity. So at this point, we were confident that at least at a functional level, these fiber types were unique. So once I had prepared them all for LCMS, the first thing that I'm showing here are the chromatographic traces. This is a base peak chromatogram from a representative fiber of each. And just kind of at a big picture level, the first 2 traces look relatively similar, whereas the third trace definitely looks unique. And this is largely due to the fact that at the protein level, there's different fast twitch and slow twitch affiliated proteoforms found within these fibers, which I'm going to show in more detail here shortly. So I think one of the most interesting findings of the paper has to do with myosin heavy chain. So I've already mentioned it. It's more or less SDS page gels were performed in the past to set like low grade 4% SDS page gels are usually used for single fiber classification based upon which isoform of myosin heavy chain is found in that fiber. So it's the motor protein of muscle thick filaments. It interacts with actin on the thin filament to convert chemical energy from ATP hydrolysis to mechanical energy. And that's really like the motor of the cell. And now, as I've said about the gels, this is how single muscle fibers are actually classified, where in slow twitch muscle fibers, they're classified as type 1, whereas fast twitch can have up to 3 isoforms, type 2A, type 2B, and type 2X. So what was exciting when we were parsing through the data outside of seeing, really large charge states, which is also exciting, was that when I was deconvoluting the data, I was finding about 4 unique masses consistently. And when we overlaid all of the fiber data of these myosin heavy chain measurements, we're actually able to do our own classification of the fibers found in our samples. So leaning on some literature measurements that show that the soleus is, about 100% type 1. We're more or less getting one measurement for our soleus samples. We're able to classify that as type 1. Knowing that the VL was almost exactly type 2B, and the fact that we were getting pretty much one measurement for the VL samples, we classified this as type 2B, which then allowed us to know in our Plantaris that this was the type 2B peak. And due to the fact that the Plantaris was the most heterogeneous one, we're able to find, based upon their sequences, that these three were type 2X, and the other peak was type 2A. Excuse me. So this, at a protein level, is showing the classification, whereas before we were showing at a functional level a similar trend in terms of the myosin heavy chain classification for the function. So that was very cool. Another thing that we showed in this paper, and it's been showed in the single cell omics field in general before, was that, from cell to cell, everyone kind of has a unique personality. But what was really interesting was being able to show this at the proteoform level. So the example I'm showing here is myosin light chain 2, which is it's termed the regulatory light chain because it's basically phosphorylation that controls the activity of this protein. So obviously that's an important thing to capture in a measurement. So you can see for the VL fibers, where VL and Plantaris mainly have the fast isoform of this protein, and Soleus has the slow 2S. But you can see for VL, the phosphorylation is relatively consistent, and the X or Y axis here is a P total value, which essentially is a measurement of the relative amount of the modified proteoform or the phosphorylated proteoform. Whereas in the Plantaris, the more kind of mixture of the more heterogeneous tissue, it has P total values that can go from 0.4 all the way down to something like 0.22. So basically, you're seeing that from fiber to fiber, this phosphorylation is actually changing. Whereas if you were doing an ensemble tissue type measurement, say of these 6 fibers, you'd probably get something that looks like something in here. So maybe for something like this one where they're relatively consistent, you might get a very similar measurement. But with the other two tissue, it shows that doing this at a single fiber level definitely captures a better snapshot of the proteoforms at that moment. Of course, if you're talking about top down, that is inferring that you did MS-MS. So to kind of show the power of doing top down at the single fiber level, we chose to align the sequences of the essential light chain. So MLC 1F, 1S, and 1V. 1F and 1, I believe 1F is in Plantaris and VL, whereas you can find MLC 1S and 1V in the Soleus single fibers. So when we align the sequences, just to give everyone some context of how similar they are, basically everything that has no color means that the sequence is shared. Anything with purple means that at least 2 are shared, and then anything that's green means that there's no homology. So if you're thinking in terms of bottom up and getting trypsin involved, you would hope that you have enough unique peptides to characterize these different isoforms. But when you do it at a top-down level, not only were we able to separate these all chromatographically, but when we perform actual collisional-based sequencing, there's unambiguous characterization of these different protein isoforms, even though they have different N-terminal modifications. and highly similar sequences, we're still able to confirm, at an MS1 level where we have high mass accuracy of the intact protein itself, as well as at the MS2 level where we're generating these sequence-specific ions. And this was used with MASH, great software, still use it. So kind of the conclusions and future directions for this work was that we developed the high sensitivity top-down proteomics method for the analysis of proteoforms from single muscle cells, which was the first time that was shown. So usual biochemical assays kind of have to dedicate a group of fibers to, run a gel on to figure out what the myosin heavy chain composition is, perform a Western blot to maybe perform some type of protein quantitation, et cetera, et cetera. The nice thing when talking to our biology collaborators about this is that you kind of get all of that information from this type of assay. We showed that there's fiber-specific isoforms of myosin heavy chain that were not only detected, but used to classify our single fibers at the protein level. And the data also showed that there's, fiber-to-fiber differences in PTMs and isoform expression in single fibers across the different muscles. So I think a future direction that this could go is trying to more connect muscle proteoforms with function in the context of some type of muscular related disease, and see what's going on at the single fiber level. And the mass spec portion, as you're going to see from the next case study, is there was nothing specific about the mass spec or LCMS method. It is generally applicable to high sensitivity experiments. So all in all, that from starting to work on it to, I think it was published like a week before my dissertation, this was probably 3 or 4 years of work. So that was really nice to see it published in a nice journal and get wrapped up. Okay, so the next case study I want to talk about is called small scale serial size exclusion chromatography, or S-cubed SEC. And before I get into this, I want to give some acknowledgments on the front end to both Lauren Eilers, who was an undergrad that worked with me for 2 or 3 years. She did all of the sample preparation for the data that actually ended up in the paper, and Holden Rogers in Ng's group, who's a co-first author on this paper. He did a ton of the data analysis, figure making, and manuscript writing to get it all wrapped up and published in Chem in January of this year. So couldn't have done this without them. But let's get into this one. So the human proteome has a wide range of proteins with different molecular weights. And as I said in the first part of this talk, anything that starts to get up to here around 50,000 Daltons gets a little tricky with top down, and specifically when it's in a complex mixture where there's smaller things there kind of suppressing the high molecular weight protein signal. Now this is not ideal because there's a lot of proteins that we care about over here. So strategies that people have come up with in the past to separate big proteins from the smaller proteins that are easier to detect, from Ng's group in particular, size exclusion chromatography has been a popular approach. I'm going to get into that more in the next few slides. Another technique known as gel-free has been used quite a bit, where it's essentially an SDS page on its side, and it elutes into the liquid phase, which is a nice high-resolution separation, but you have to deal with SDS, as well as more recently PEPIMS, which is a lovely acronym. But basically, that's again using an SDS page gel, getting the proteins out of the gel, and then so that you have your size-based fractionation, which then allows you to start getting after some of these larger proteins. So as I mentioned, this work was inspired or built off of whatever you want to use. Tricia and Veggie's work, where they first termed serial size exclusion chromatography. So essentially what they did was they took two 9.4 millimeter SEC columns that had different particle attributes, mostly pore size and particle size itself, and then connected them serially so that one, the path length was longer, and then also the actual chance to perform this size-based separation, you have two different particles to interact with the protein molecules. And they showed that this increased the resolution of their SEC separations. They were able to collect fractions and perform online reverse phase chromatography top down. And it was a great paper. You can see from, they actually connected three, excuse me, just in here it was two, but they connected a 1000 angstrom SEC column with two 500 angstrom columns. And you can see from this gel that they have a really nice size-based separation. And then when they're able to do online reverse phase coupled to mass spec, you can see the detection of some of these really large proteins. But one of the challenges with this work was that it was pretty low throughput because they were, on the offline SEC separation, it was a 500 microliter per minute flow rate. They were collecting 500 microliter fractions, 1 minute long fractions. They were loading a lot of sample from a large amount of human sample. So kind of a simple idea that Tricia first started and I picked up once she had left was just to take the 9.4 millimeter column and move down to a 2.1. And this has some pretty significant ramifications. Now you're going to a 29 microliter per minute flow rate that collects really tiny fractions, and you're able to theoretically use a lot less sample from a much smaller piece of tissue. So that was kind of the idea. And then as that was also happening, I was also working on this high sensitivity LCMS end of it. So we kind of thought, why not put one with the other and see what happens. So the first thing that we did was kind of screen different SEC columns. As I mentioned, they're 2.1 millimeter in inner diameter. We showed that, one SEC column wasn't, because basically if you calculate the volume, if you treat a column like a, it's basically the volume is significantly less when you're going from a 9.4 millimeter column to a 2.1 because the radius is so different. So essentially, the one column SEC separation wasn't good enough, but when we connected to a 1000 angstrom column with a 300 angstrom column, we actually see a much higher resolution and detect all of these standard proteins that were in our mixture. So another thing we had to kind of play with was that since we're starting with a really small piece of tissue, this is one of my favorite figures ever. It's in the SI. But basically it shows you how small this piece of tissue is that we're trying to do, multi-dimensional top down on. So we couldn't follow like our normal like protocol for tissue-based extraction of the contractile proteins. So we more or less scaled everything down on the sample prep end using, the smallest amount of volume of extraction buffer as possible, handling the sample as little as possible, etc. And what we see when we perform this separation is more or less two main peaks by SEC. This kind of shows you the concentration of protein in each of the fractions. And then you can also see by the SDS page that we are getting a size-based separation. Maybe not as high resolution as Tricia's, but again, the amount of stationary or stationary phase, our particles in the column itself is significantly less, so that makes some sense. So essentially here is the first figure of the paper, kind of going through the entire workflow, where we're starting with 1 milligram of human heart tissue, performing a two-step extraction, loading 20 micrograms total of protein onto our offline serial small scale SEC separation. We're collecting the eluent directly into an HPLC vial, at which point we were loading about 150 nanograms of protein per fraction. So this kind of shows all of the gel in which I already showed, and then the chromatographic trace. And kind of the big thing that I took from this is that when you look at the 1D separation, so no SEC was performed, and compare it to some of the later fractions, they actually look more similar because you can tell that it's the smaller, higher abundance proteins that are mostly being detected in the 1D separation, whereas in fraction 1 and fraction 5 with some of the higher molecular weight proteins, there's actually a pretty unique chromatographic trace in comparison to the later ones. Now, to kind of show some examples of being able to detect large proteoforms from, 1 milligram of tissue, one of the first ones that I noticed was, so again, the black trace is just the one-dimensional trace where there was no SEC performed, and the red trace is the trace of the first SEC fraction. When you average the no SEC one, basically you see the existence of this 32,000 Dalton protein, which is tropomyosin. But when you do the same thing on our fraction 1, now you have a completely different protein that is about 150 kilodaltons in size. Similarly, in this later stage, when you average the spectra from 31 to 32 for the no SEC one, you pretty much get a pretty nice spectra of actin, which is a 42 kilodalton protein. But again, when you're averaging the same exact space on the first fraction, we're now getting the cardiac isoform of myosin heavy chain. So outside of, the really big proteins, we also showed the benefit of, more medium-sized proteins, so between 50 and 100 kilodaltons. Similar analysis as to what was just done in the previous slide, but again, for the no SEC trace, you see the existence of troponin C, which is a pretty high abundance and low molecular weight protein. But when you take the third fraction, so fraction 3, probably some of these proteins very faint here, in average, you kind of wipe out the troponin C and ATP synthase subunit, and now you're seeing the 71 kilodalton protein. So that I was just for time, I only wanted to show the main figures of that paper. There's also some good stuff in the SI. Holden did a great job wrapping it up and doing a ton of work for it. So thank you to him again. But we basically showed that our S cubed SEC high sensitivity LCMS method is able to detect high molecular weight proteoforms from a really complex mixture in a really small sample to begin with, by providing that additional size-based separation that you don't typically do in a 1D approach. Again, similar to this previous paper, we use this to study big proteins from heart tissue, but more or less the method itself is amenable to other sample limited case studies. So, small amounts of cells, things like EVs. I think this approach is broadly applicable. So I don't want to ignore some other really good papers that are happening in this space as well. There was one in 2020 in Chem from the PNAS group using the nanopods to do top-down. The Ivanov group had a really interesting one in 2022 where they're more or less doing their single cell or top-down proteomics all within a capillary. And the Kelleher group has a really interesting paper that I'm guessing is in revision somewhere, and is currently on bioRxiv, where they are kind of using an approach to create these really quick ways to do single cell proteomics, using DESI, I believe, getting their cellograms and single cell spectra. There's a lot of good stuff in this paper. Okay, so to kind of wrap up, I think there's things that can be learned from the single cell bottom-up proteomics field. And that is clear that there's a very small amount of sample that we're beginning with, and everything that's in bottom-up proteomics is going to be trickier in the top-down world. So really, there's major themes that have been seen in these papers that say minimize your sample handling, use the highest sensitivity chromatography, ionize as much of the sample, get as much of the sample ionized into the mass spec as possible, as you can see by things such as the recent timsTOF pros are all kind of with bright ion sources to maximize the amount of ions that they're detecting. The ways in which the MS acquisition was actually done can boost sensitivity, and then a lot of this will come at the back end, and that this data is going to be unique to our bulk proteoform measurements as well. So there's a really fun little review that Ryan Kelly wrote in MCP, where he has this little recipe card for single cell proteomics. And I think while this is taking a more peptide-based approach, all of these things are going to contribute to as this field advances as well. I think it's really exciting, and I think as it goes further, it's going to be hardware and software that continue to enable this field, and people like people in this community that will really drive it. So with that, I'd like to give some acknowledgments specifically to Ing, great PhD advisor, everyone in the GA lab. I miss you guys. It's been about a year since I've been gone. For the single fiber project, Kyle and Yutong, the S-Cubed SEC project, Holden and Lauren. Both of these works, Andy Alpert was instrumental in, and I hope to see a lot of people at ASMS coming up in June. I'll be chairing the session that's actually in memory of Andy, so it'd be great to see a lot of familiar faces there. The Nuomics people, our team, specifically Dao Jing, as well as Bruker and all the funding that I had. And again, thank you for giving me the opportunity to talk here. I've always really enjoyed these Proteoform Thursdays, and I'll take any questions.
Great, thanks, Jake. Fantastic stuff. It's great to see the evolution, and hopefully it continues on an upswing like that. So we do have a couple questions to start out in the chat here. Jeff Silver had a question about the use of affinity reagents, how they can be useful in top-down for discovery.
That's an interesting one. I think definitely something that is important. So something that Ing's group and the group that I came from used is like nanoparticles to do affinity enrichment. There's other approaches that use biotin and things like that. But again, this is like solving the problem or challenge of trying to get your protein of interest away from everything else that's going to get in the way of not only detecting it, but having enough of it to do MSMS and get good sequence coverage itself. So I think it's currently something that people use and will continue to be really important. I think one of the challenges there too is that you kind of need to know what your target is, where you're kind of saying here for discovery. I think that's space that can continue to be developed. Of course, you can get a protein itself, but it's these different molecular forms that is going to really matter. But yeah, it's a really good and interesting question.
Yeah, great, thanks. And Carolyn has a question about how time-consuming is the one-pot sample prep? Is the first question.
Yeah, that is a good question. It's actually pretty fast. So basically, the time-consuming part is like getting the fiber into the actual tube. And then you basically have to circle the fiber immediately because then you won't know where it was. But once you have that all done and you take your fibers to your own lab and have it in the freezer, you basically add the HFIP uh, and do the freeze thaw, and then basically you load that whole entire thing onto the column. So I think the sample prep part only takes, I'd say, less than 2 hours, which is nice.
Okay. And the second quick question is the pneumomics source can be connected to any brick or instrument. I'm not sure if that's in your
Yes.
wheelhouse or not, but
Yeah, it can. Something that, because the timsTOF, I know for sure it can. I think that would be really interesting to do. It because basically the timsTOF has the same front end as the Maxis and Impact, which is what I previously used. I'm not sure if it would work on their FTICRs, but I don't know why not. I think it all has very similar source dimensions, but that definitely on the timsTOF, and I hope someone does that soon.
great, thanks. And a question for Ronaldo. Can you mention the main optimizations performed in the TOF instruments?
Yeah, so that's a good question. Mainly it was in terms of the proteins that we got into the mass spec, we were making sure that we spent as much time as needed on the MS2 acquisition and tried to boost sensitivity more by getting more MS1 scans. So essentially getting enough coverage to say that's that protein, that's that specific isoform, and then we could really try and boost, yeah, sensitivity by having more and more MS1 scans.
Okay, great. And his final question what deconvolutions have you used for the big proteins?
Yep, another good question. So we use the maximum entropy deconvolution and just had to set the resolution really low to something like 10,000. So you definitely on the deconvolution itself, you lose like the isotopic pattern, but it was trying just the fact that it was deconvoluting like plus 260 um, positive charges, it gets a little crazy. We also played around with Unideck a little bit. I think there's more that could be looked into there, but it yeah, it also seemed to struggle a little bit with the really big charges.
Okay, great. And Colleen has a comment in the question. A lot of single cell high sensitivity bottom-up methods have now been fully automated to address some of the challenges you mentioned. Do you think some of this automation should be adopted to top-down applications in lieu of just trying to continually add optimizations to specific parts of the top-down workflow?
Yeah, very, very good. So I think part of the reason why the bottom up field's been able to do this is because there's like a discrete number of steps that have to be done. You have to, denature, you have to reduce, you have to alkylate. So there's commonalities, and you can kind of, the sample prep itself is, I feel like, more uniform. Like you add something to lyse the cell, and then from there you're able, to clean it up with the trap column for the bottom up field. Whereas I think that's tricky right now with like where we're at in that don't think there's as much of a like common workflow that could be implemented that automation could, deliver to many different places. I think it'll definitely be a huge part of it because we are human, and at these levels, it's really difficult to reproducibly and reliably do a lot of these sample preps. And I mean, I would have tried to use it in even smaller volume, like nanoliter volumes, like the nanopots team's been able to do in Cell in One and all these things that you're referencing. But I just made it as small as like I could do like with my own hand. So I think what you're getting at is definitely something that's gonna like solve a lot of the challenges that face this field.
Sounds good, Jake. And Jilu has a question. Can you imagine any part of this technology could be applied in biopharma?
Yeah, that's really interesting. Um, I know that, so I've thought about it in terms of cell culture a lot because our cell culture, even like bioreactor to bioreactor, can be like the cells show kind of some very heterogeneous behavior. And while this has kind of been noticed at like a single cell peptide level, it really seems like it's post-translational modifications and things like phosphorylation that could, there's a lot of crazy signaling going on, it seems like. So I think, yeah, definitely like to have a better understanding of like cell line development or even like clone selection, if you could do, single cell top down at that level, I think it would be really something that people would be interested in. Um, yeah, that's something I'm definitely going to think more and more about.
Great, thanks. Any other questions for Jake? I just have one final question. If you look at the workflow and your whatever, however many steps, if you had to invest in just one of those steps to improve it, which would be your number one investment?
Yeah, that's a good one, Paul. I think sample prep. I think that sample prep is where I probably lost the most sample. I feel like it's not the mass specs anymore. I think the mass specs are pretty darn sensitive, especially some of the more recent technology that's come out of Bruker and Thermo. The mass spec is very sensitive. It's yeah, dealing with the front end and actually getting your sample onto a column. I think that that's where the most improvement can happen.
Great. That sounds good. So I think we'll close out there. Jake, thank you very much. This is fascinating stuff, and we look forward to more developments in this arena. We really appreciate your effort and sharing all this with us. So
Yeah,
thank
thank
you,
you for having me.
and we wish everyone a happy and a nice day. Thank you. Goodbye.